Okay, today we're going to be talking about a lab that we're going to be doing next week, where we're going to test the quality of the water behind South Ave. That stream is called Leggett's Creek, and we're going to be using the organisms that live in there to see how healthy the water is. So go ahead and write down this title. We're using these creatures called macroinvertebrates, and we're going to be taking a look at what those exactly are as we go through this, and that's what we're going to be doing with this lab. Okay, here's some vocabulary that you need to write down. So take a few minutes and get these words down. We're going to be using a microscope called a dissecting microscope. Dissecting microscope is one that it's not like the ones we've been using. They don't magnify as much. The ones we'll use, they'll magnify about 2 times to 4 times. And they're really just to see the outer surface of a creature. The creature can be alive. It can be something bigger. So it's almost like a fancy magnifying glass. We'll be using some magnifying glasses too. What we're looking at is called indicator organisms, and they're indicating something to us. And what they're indicating to us is, are they there or not? And if certain ones are present, then that tells us something. And if certain ones are not present, that'll tell us something else about the quality of the water. And finally, this word I keep using is macroinvertebrates. A macroinvertebrate is an organism that has no backbone. That's what invertebrate means. Macro means you don't need a microscope. Now, we're going to use microscopes and magnifying glasses so we can see some features of them. But they're going to be mostly like kind of like larvae, mosquito larvae, dragonfly larvae. The creatures that are going to go through a metamorphosis and become a mosquito, or they're going to become a dragonfly. Those are the kind of creatures we're going to be seeing. Some kinds of aquatic worms, some beetles, things like that. Okay, here's some more things you have to note. We said this already when we studied these larvae. Remember, larvae is almost like a caterpillar. They're the certain form of insects that are going to go through a metamorphosis to become something that probably flies, but they live in water during the first part of their life. We're going to see some of those. We're going to see that some are sensitive to pollution, some can tolerate low levels of pollution, and some thrive when the water is heavily polluted. So based on what we're seeing, that'll tell us how polluted the water is or how clean the water is. And these creatures are low on the food chain. So they're going to be herbivores, maybe secondary consumers. Those are the kind of things that we're going to be looking at. So take a minute and jot all those notes down. Okay, finally our lab. I'll give you some more details next week, but I'm going to bring in a water sample on Monday. If not Monday, we'll get to it Tuesday. And we'll be looking at the water sample for a couple of days because we want to catalog every single critter that's living in the sample I bring in. And we said this already, based on what we're seeing and what we're not seeing, that will tell us the quality of the water. I don't think you need to write these down. I think you've already written down stuff like this. Okay, now start writing again here. We're going to calculate something called the pollution tolerance index. This is a number that we go through a whole calculation sheet based on what we've been seeing, and that will give us a number that will finally determine is the water good quality, fair quality, or poor quality. So it's based on what we're seeing as a whole class as we keep track of everybody's data. So write that down. Once we determine this number, it'll tell us the quality of the water. So take a minute and jot those two down. Okay, we'll look at these things as we get into them next week. Okay, you don't need to write this stuff down. But what you're actually expected to do next week is you're going to go get a sample out of the water samples I bring. I'll show you how to do that. You're going to use dissecting microscopes, magnifying glasses, trying to get an idea of what these creatures are. I'll give you these laminated maps. There'll be like these keys that we can use to figure out what's what, so you know what to write down. And we'll just be doing that for a couple of days, and then we'll start getting into the pollution tolerance index aspect of it by Wednesday or so. Alright, I found this picture online. I probably could have drawn something this good, but this is what I'll be doing. Okay, so I'll be in Leggett's Creek Monday morning before school, and as the water flows from right to left, I'm going to put something called a kick net downstream from me, and I'm going to scuff my boots up on the bottom where all the rocks are, and what that does is it frees up all these insect larvae that are hiding under the rocks, and the stream washes them right into the net. And then I'll put it into a bucket, I'll bring it in, and we'll check it all out. So that's how I'm going to go ahead and get your sample. Okay, last thing I want you to write down. What is the advantages of using macroinvertebrates that are present in the water versus, I don't know, if you have like a swimming pool at home, you just use a chemical test kit. It tells you the chlorine, the pH, things like that. What would be the advantages or disadvantages to doing this over just chemical testing? So I'm going to make a little side-by-side chart here. One advantage of using the macroinvertebrates is they've been living in this water long term. They've been living, reproducing, eating, getting eaten. This is their habitat, excuse me. So we really get a good picture of the long-term data, the long-term health of this body of water. Chemical testing gives you the levels at that moment. We'll do some chemical testing for nitrates and nitrites and ammonia and things like that. But that's only valid at the moment you do the tests. So using the macroinvertebrates gives you the advantage of you're seeing what's living and not living in there. Chemical testing gives you precise numbers, but they're only for that period of time. Macroinvertebrate studies take a lot longer. I have to go on Monday. I'll get you the water sample. It's going to take us a couple class periods to get through it. It just takes a lot more time. If you've used chemical testing for fish tanks, swimming pools, those sorts of things, you know it takes minutes. Okay, so it's very fast. A lot more equipment with the macroinvertebrates. Like I said, you have to get a net, a container to put them in, microscopes, magnifying glasses. It is a bit more involved. The chemical tests are cheaper. A lot of times it's just a stick that you dip into the water and you see if it changes the color, or you put drops of some kind of indicator solution into the water and see if your test tube turns pink or blue or whatever. So it's a lot cheaper. I think the macroinvertebrates is a lot more fun. I think you guys are really going to like it. And that's why I wanted to use this video, just to introduce this to you today. And then next week we can kind of jump into it and I'll show you everything else you need to know as we're going on it.